Welcome to the channel. In this video we are going to be considering 100 genetics terms. You see, genetics is a branch of biology that has its own jargons, which you must understand in order to have a good conceptual grip of this branch of biology. So we're going to be starting with the first 30. Now we are starting with 30 because of the limitedness of space. Genetics is the first one. Genetics is the study of, that's on the technical notes, is the study of variation and heredity. Now what it basically is, etymologically, genetics is gotten from the Greek word genesis. And you know that Genesis means origin. Genesis means origin or beginning. Talk of the Bible, origin, beginning, all right? And that origin is actually gotten from the Greek word genia, which refers to descendants or a generational, descendants of a particular ancestor. So, like people, or organisms, successive organisms in a particular, what they call genealogy, that arise from the same ancestor. All right, so that's what genia is. So, genetics basically study how characteristics are being passed from the parent ancestor down to the offspring, and the same characteristics propagate until even future generation. So that is what genetics studies. That's why those two branches are called heredity and variation. Now heredity meaning how traits are transferred, and variation meaning the differences that now occur, because of course that it is known that characteristics are transferred, but it turns out to be that the offspring that is formed or progeny is not exactly the same as the parent. So because of this reason, we know that there is a variation. So we want to study what is the cause of that variation. So that is one of the things that genetics studies. All right. So I said heredity, which is the second term, is basically the study of how characteristics attributes are inherited by offspring or progeny from their parents. So of course they are based off of chromosomal transfer that happens in the cellular level during fertilization and the rest of that. So where those characteristic traits are transferred from the parent to the offspring is heredity. Then variation is simply the fact that offsprings do not look exactly like their parents. Of course they look like their parents in some sort of phenotypic level, but do not exactly have the same traits as the parents. So this variation in the traits even that they have inherited is what we call variation. These differences, in fact, the best term that is synonymous to it is differences. So that's what variation studies, right? The next term is DNA. DNA basically stands for deoxyribonucleic acid. Nucleic acid. And what that means is that, you see, look at the word deoxy. Deoxy means that the ribose sugar, the pentose sugar has been deoxidized. So because of it has lost an oxygen atom, it is called deoxyribonucleic acid. All right. Now it is basically the structure that codes for life. Basically, that is what DNA is. It's a double helical structure that has the code in which everything about an organism is designed after. Now, it carries a code. The DNA carries a code that which is interpreted into what the organism becomes. So that's what the DNA is. It's the basic unit in which it is the unit in which life character of every living organism is imprinted on. All right. So then of course there is also the RNA. So I'm going to be treating DNA and RNA. RNA is basically the ribose nucleic acid. All right. The basic difference between DNA and RNA is that The DNA, first, there's a difference in their structure, and of course, there's also a difference in their function. You see, the DNA basically codes for general characteristics of the organism, general characteristics, or sometimes even specific characteristics, actually. But I mean, when we talk about the integral part of the DNA, of course, it codes for specific characteristics. But the RNA codes for specific proteins. So RNA is for protein manufacture, and of course some degree of regulation of biochemical processes. But the DNA basically is a structure that codes for life. So another basic difference between them is that the RNA imitates the DNA for its own structure. So the RNA doesn't possess a structure except it copies the DNA. Alright, so and it is called ribonucleic acid. So the ribose sugar is still complete at here, but here it has lost an oxygen molecule atom. Alright, what is a gene? Now a gene is basically the word genia that I talked about. A gene is also what we call the Mendelian factor. We call it the Mendelian factor. Of course, over the course of the study, we are going to know that there are some other genetic characteristics that are non-Mendelian factors because of transferability that has to do with the other exogenous factors, that cannot be factored into a Mendelian crossing. All right. So gene is basically the basic unit of hereditary characteristics. So the smallest unit for a trait, the smallest unit that codes for a trait, a particular trait in human being. For example, the smallest unit that codes for my skin color is called a gene. The smallest unit that codes for my height is called a gene. So a gene is basically the smallest unit of hereditary characteristic, and it is simply a segment of the DNA sequence. It is simply a segment of DNA sequence, the gene. All right. Now, allele. Allele is actually gotten from is diminutive of the word allelomorph. 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 Of course, allele still meaning allele. That is the diminutive form, and morph means form. All right. So, an allele is gotten from the Greek word allelon. Allelon, which means mutual. Allelon which means what? Mutual. So this brings us to how the understanding of what an allele is and how it acts. So it's basically the alternative form of a gene. So if a gene, for example, the gene that codes for height in human, for example, if we use T to represent the gene for tallness, and we use small t to represent the gene for shortness. So now this individual that has this big T and small t is going to be tall phenotypically, provided that the tallness is dominant over, I'm going to explain those terms of course later on. Now, is dominant over this. But what I want us to understand is that these two forms are genes. So, but when we talk about one with respect to the other, we mean that one is an allele of the other, meaning that one is the alternative form of the other. For example, black and white. So, well, they may not necessarily be opposites of each other, but basically here we need they are opposite of each other because they are alternative forms. So it's either you are tall or you are short. So, but both of them are coding for the same trait. And what is the trait? Height. How that is clear? So there is the gene and there is the trait. So this gene is an allele of the second gene, which and both of them code for height of the organism. Alright, so that's an allele of the alternative form of a gene. Then the chromosome. So basically, I'm going to be explaining the chromosome, the chromatid, and the chromatin. You see, chromosome is gotten from two Greek words, and they are chroma and soma. You see, chroma meaning stained, while soma meaning body, from somatic cell. Okay, so stained body. So the reason why they are named after that is because I've actually forgotten the scientists that named them first, but it was around Morgan or Bates. One of them named them first, and he named them because of their characteristic property of being able to be stained easily during the metaphase stage of cell division. So you will notice that, no, not you will notice, of course. If you have gone over that video I made on meiosis, you hear me talk about the fact that chromosomes are more conspicuous during the metaphase stage. So during this metaphase stage, they are easily seen with even a light microscope because they are conspicuously stained. So that's why they are called chromosomes, because when you stain them with dyes, specific dyes in the laboratory, they exhibit peculiar colors that makes them very visible. All right, so that's why they are called chromosomes. But I've not even explained their difference. Now, their meaning. Now, a chromosome is simply the segment. A chromosome is simply DNA wound up, or coiled up. Chromosome is simply DNA that is coiled up severally, to the point that we say they are condensed. So they are coiled up severally on each other, and through the help of proteins that we call histone proteins. I'm going to explain the meaning of the other function of histone proteins, so I'm going to talk about it later. But basically know that the first fundamental function of that histone protein is to allow DNA to coil up. Now, when we have a complex, this is the difference between the three now. When we have a complex of just the DNA strand and this histone protein alone, not coiling on each other, we call it the chromatin. So during the interphase stage, which is the predivision stage, the stage before that precedes division, cell division. Now, during that phase, when the cell is not ready for division, what we see is called the chromatin material. So you just see thread-like structures scattered around the nucleus. If this was the cell and this is the nucleus, I said this is the nucleus, you will see the chromatin materials scattered within the cell. So these are going to be chromatin materials, or the uncondensed form, the uncoiled form, the unwound form of the DNA molecule, the DNA strand. Alright? And now, a chromatid is now this. We have the common way, the common representation of a chromosome is this shape. Now, and we know that we call this homologous chromosomes. Homologous chromosomes. And why are they called homologous chromosomes? Because they possess the same type of gene at the same locus, so they are alike, so they pair. Now, but basically, my point is this: when these two guys detach at the anaphase stage of cell division, we are going to have something like this. Now, this point, there's a name for this point, which is called the centromere. Actually, I'm still going to talk about it. Now, this, when they now separate, this guy here is going to be called the chromatid. Notice, even at this stage, it is coiled up, only that it is separated from its sister. Now, this is the stage in which they appear having coiled up from chromatin. So when chromatin and histone protein coil upon each other, they form what? Chromatid. Now a chromatid can also be called a chromosome. A chromatid can also be called, but a chromatin is not a chromosome. All right. So chromosome. So it's just like chromosome. Chromatin is just basically chromatin plus histone protein gives chromatid. So, chromatid is synonymous to chromosome, alright? While this common form is homologous pair of chromosome. Alright, so that's the basic difference. Alright? Now, we get to the next one, which is the haploid. The word haploid is gotten from the Greek word haplous. And haplous basically means single. Single. Now, what does single mean? Why is it called single? You see, an haploid cell or an haploid organism is, of course, it's basically a cell. Is a cell that possesses half the number of genetic characteristic of the parent. Now, there are two types of the cell division. We have the mitosis, which is the mitotic cell division, or the meiotic cell division, which is meiosis. Now, in the mitotic cell division, the cell usually gives rise, if I'm going to use the concept to explain both already. In mitotic cell division, the division happens such that the offspring or daughter cells that are given rise to have the same exact number of genetic characteristics of the parents that gave rise to them. So, if the parents were diploid, of course the parent will be diploid, so they are going to produce a diploid organism. So diploid is represented by the number, by this 2n number, and this one is represented by n. So showing that when the offsprings are formed, they have the exact same number of chromosomes as the starting parent. While in the haploid, they have only half the set of chromosomes that the parent possess. Alright, so that in human, we have 46 chromosomes, which is equal to 2n, which is our 2n, our diploid number. So showing that n is going to be what? 23. So it means that our any aplontic cell in our body, of course, if we are an haploid, it will be called aplontic. So any aplontic cell in our body is going to be what? It's going to have 23 chromosomes, not the pair, just 23 chromosomes. And I'm going to tell us the implication of that later on. Now, and of course, the diplontic cells of our body, which are the somatic cells, are going to be going to have 46 chromosomes, which are 23 sets, 23 pairs. All right. So, and the aplontic cells of our body are basically non-somatic cells, which are the sex cells or gametes. All right. Now, then the meiotic division, I said this one is a division that gives rise to exact same number of an individual that has exact same number of chromosomes as the parent, while meiotic is the one that gives rise to an individual that has only half the number of chromosomes that the parent has. Now, the implication of this meiosis, the reason why the daughter cell that is formed has only half the number of chromosomes that the parent has is because of this. The kind of cells that engage in meiotic divisions are cells that must recombine with other cells to form a new organism. That is, they are gametic cells. So if they are gametic cells, it means that there has to be a syngamy. There has to be a syngamy at the end of the day. Syngamy means a fusion, which means a fertilization. So now there has to be a fusion of two nuclei. If those two nuclei fuse together and they were aplontic, it means that they are going to double the number of chromosomes that were in the parent. That is, provided that the cells that were produced, the gametic cells that fused together, were actually diplontic. It means diplontic. It means that they are going to produce a zygote or an organism that is now what? Tetraploid. I hope that is clear. So it means that they are going to double the number. So if there are 2n plus 2n becomes what? 4n. So, and it's going to propagate for each successive generation like that. Because if the next daughter cell has 4n and then comes with another 4n, then it means that the next generation will be 8n. And then number of chromosomal number keeps increasing and propagating like that. So that is the implication of this meiotic division. It has to be half, so that at each point of fertilization, we keep producing only a diploid number. Alright. Now the next is the zygote. Of course, I've talked about a zygote just now. The word zygote is gotten from the Greek word zygotos. Zygotos basically means joined, or agreed, or fused. Joined or fused. Alright. Now, a zygote is simply the organism that is formed from the fertilization or syngamy. The word syngamy is very simple. It's basically the technical biological term for fertilization. So the zygote is any organism that is formed from the syngamy of two gametic cells. Alright. So syngamy is gotten from two Greek words, which are syn and gamy. Alright. So you know, syn means together, to agree. Alright? While gamy means to marry. Remember these words polygamy, monogamy, those words. So that's where they are gotten from. This word they are gotten from. Gamy means marriage. Marriage. So it means syngamy, coming together to marry. So it's the technical term for fertilization. So when the two nuclei, the gametic nuclei, fuse together, we said they are fertilized. Okay, right. Now the next is the dominant gene. Now we talked about allele just now. Alleles we said are the alternative forms of gene. So if I have a gene like this, since we are done with this, I'm going to clean on the board off here. If we have a gene, this gene that codes for tallness, and we have another gene T that codes for shortness, when is the organism going to exhibit this? When is the organism going to exhibit this? Now, this big T, now listen, it is an arbitrary, sometimes we pick arbitrary letters, all right, to represent our genes. So here we are picking T, and usually it is a common practice that it is the dominant allele, the dominant gene or allele. How that is clear? It's also allele. So it is the dominant gene or allele that is written in capital letter. So now, the fact that it is called dominant doesn't mean that the recessive, it has killed the recessive. Alright, but I'm going to explain that later. I'm going to explain that further. Let me explain what dominant means first. Dominant means the gene that expresses itself over another, that expresses itself in the presence of another gene in the phenotype of the organism. So, if tallness is seen in the physical appearance of the organism, of the human, then it means that the tallness is dominant over the shortness. But if otherwise, it means that other trait is dominant over the second allele. Remember, they both code for height. Yes. So now if the person is tall, appears to be tall, at the end of the day, it either means that tallness is dominant over the shortness. And if the either is short at the end of the day, it means that it either means that an individual is the shortness is dominant over the tallness. Yes, there is a possibility that the shortness is dominant over tallness. It is not only in the recessive form. Now, recessive genes are genes that do not express themselves in presence of that gene until they are homozygous. So, except the individual is homozygous for these genes, the genes cannot express themselves in the phenotype of the organism. Now, the fact that they are recessive does not mean that the dominant gene totally kills them off. Because from non-Mendelian factors, we begin to understand that for example, tallness in humans is a polygenic trait. It is not a trait that is, I'm going to talk about polygenes too. Now, it's a polygenic trait. It's not a trait that is dependent on only the genetic, only the gene that carries the trait for tallness or for shortness. No. So this is a polygenic trait. So we cannot just say that it's based off of, say, the dominant tallness and the recessive shortness. No. But fundamentally, just know that the dominant gene is the one that expresses itself in the phenotype, while the recessive gene is the one that doesn't express itself until the individual is homozygous, TT. So, but the dominant allele, if the individual is homozygous and heterozygous for the tallness, for that dominant allele will be expressed. So that is the name of dominant allele. Alright? Then the next one, phenotype. Phenotype simply means the physical appearance, what you see in the physical appearance of the organism. So if it's for tallness and shortness, we are still talking about, if the individual is tall, then the phenotype is tall, right? Then the genotype simply means the type of gene that is seen. Okay, this is best explained using a test, a crossing. For example, if I cross an heterozygous tall individual and a homozygous short individual, now let us produce their gametes. Now it is very important that before you cross, you produce their gametes. Produce their gametes. So these crosses to this and crosses to this. While this crosses to this and this crosses to this. So we have this guy produces T short t, big T short t, short t short t, short t short t. So basically what we have here in the phenotype is that, remember, this is dominant. So we are going to see this individual, this first offspring. This is called the first filial generation. I'm going to explain that later. First set of children, alright? So the first person is going to be, this individual is going to be tall. Phenotypically, you are going to see the person tall. This person is also going to be tall, while this person is going to be short, and this person is going to be short. But notice the genes are different. This is homozygous, this is heterozygous tall, and this is homozygous short, and this is homozygous short. So the phenotype here is going to be 2:2, and the genotype as well is going to be 2:2. The genotype is 2:2 because we have two T's, two short T's coming twice, and one big T and small t coming twice as well. So that is the genotype. Alright, the genotype, the type of gene, that's the name. Alright. Then homozygous simply means, I've talked about it. Now this is heterozygous and this is homozygous. So this one is called homozygous short. Homozygous simply means homo and zygous. So that is the same. Homo means same. So similar. So if the individual carries the same set of gene for the particular trait, then the individual is homozygous for that trait. So for example, if I am AA, it means that I am homozygous for this trait. For example, this AA is usually used to code for either our blood group or our skin pigmentation. So here, where the capital A will mean that the skin has melanin, right? So this person is going to be dark in complexion, or maybe not dark, but it's just going to have enough of melanin, right? So, but basically what I mean is that homozygous means the individual has the similar genes for the same trait, and heterozygous means the individual has different alleles. For example, this is heterozygous. Alright. Now, I've explained this and this. Now, the next one is autosomes. Autosomes are the cells that are the cells of the somatic cells of the body. They are the chromosomes, rather, of the somatic cells of the body. So chromosomes that belong to somatic cells, which are the bodily cells, or that part of the body, apart from the reproductive organs, the reproductive cells, all those chromosomes are called autosomes, while the sex chromosomes are called allosomes. The sex chromosomes, alright? So then the next is the centromere. Now we're getting into what the chromosome is. This is talking about the centromere. Now, basically, this is the very common structure that we use to represent a chromosome. You see, there is a portion here that these two chromatin strands join together, and that is the portion that is called the centromere. You see, the word centromere is basically gotten from two Greek words, which are central and mere. So central meaning center, of course, central, while mere means unit. So the units are the center of these two chromatid strands, right? Or sister chromatids, as you might call them. So centromere. So, but basically, what is the centromere? The centromere simply means is simply a portion of these chromosomes, these two chromosomes. The portion of those two chromosomes, remember I told you can use chromosomes, you can call them chromosomes as well. The chromatids can be called chromosomes, but not chromatins. Alright. So the portion of these chromosomes where there is higher level of compactness of the DNA coiling. The DNA coils so much and become really compact at this point, called the centromere. Alright, and it is for different reasons, for genetic regulation, gene regulation, and of course to hold chromosomes in place for division. Yes. So, and those DNA that are called up, compacted there are called satellite DNA, but we'll get into that some other time. They are called satellite DNA, and it's actually because of when they are separated, they form a complex that's called, that looks like a satellite. So that is why they are called satellite DNA. The DNA found to wind up compactly in this portion. All right. So there is also the centromere. The chromomer. The chromomer is basically the portion of, now when the chromatin begins to wind up, there are some portions of higher condensation, just like this centromere. There are some other portions in this body, in this portion are called arms. I'm going to talk about when I talk about chromosomes again. These portions are called arms. So in these arms of the chromosome, there are some portions that are more compact than the others, and that's because of the presence of histone proteins there. So the presence of histone proteins there are also for different reasons, of course. So the presence of those that histone proteins, they make those structures appear more compact than the other body, and they appear like bead-like structures. So, and they are called chromomeres. All right. And they also stain more conspicuously than the other portions of the arms of this DNA, right? Of the chromosome. Pardon. Then the telomere. The telomeric regions are basically regions at the end of each chromosome. So these regions are called the telomeric regions. Telomere. Remember, telomere means like the end. So the telomeric region, we talk about the end of the chromosomes. Then recombination. Recombination is another word for recombination is another word for crossing over. Now, what crossing over is the exchange of gene at a portion at different loci on the chromosomes, on sister chromosomes that come together at the participating stage of the first meiotic division. Remember, please go and watch that video on meiosis if you have not, in order to be able to comprehend this terminology very well. Crossing over basically happens at the pachynemma. And at the pachynemma, what happens is this. Remember that prior to that is the zygonemma. And at the zygonemma, what happens is sister chromatins pair. Sister chromosomes, rather, they pair. Now they form homologous pairs. Now, after pairing, what happens? This complex is called the synaptonemal complex, of course. Now, after pairing, what happens? In the participating stage, this recombination now happens, which is basically the exchange of the genes at the same locus. So let's say this chromosome comes from the father and this chromosome comes from the mother. The exchange of genes at the same loci here, or loci, the exchange of the gene at those loci between both parent chromosomes is what you call recombination. And the implication of this is this, of that process is this, that the individual that is now being formed, that is carrying one of these characteristics, is going to be different essentially from the father and from the mother in a way. So that when they separate eventually at the anaphase, you know that they're going to separate at the anaphase, they don't return the same way they came together. So they came together as sister chromosomes, they leave as non-sister chromosomes. Why? Because the parents, at different loci, the parents' genetic materials have been exchanged between each other, and that is what is called crossing over of gene. Crossing over of gene. So that is what one of the major factors that leads to variation in organisms, right? Then synapses. Synapses is basically a process where those sister chromatids come together, and that is at the zygonemma. When they come together, we say they have synapsed. So the essence of this synapse is so that this recombination can happen, right? So that when they are now separating, they separate as non-sister chromosomes, right? So It happens at the zygotine state or zygonema. Please go and watch that. I keep emphasizing, go and watch that video on meiosis. Then disjunction. Disjunction is basically what happens at the anaphase. So after they have synapse and exchange of genetic material through crossing over or recombination, I forgot to mention this. When by the time they will be living, okay, yes. So at the anaphase is where they now live. So that process of living is called disjunction. So when they now live, they are called recombinant chromosomes, and those genes that are exchanged are called recombinant genes. So they are recombinants. All right. So disjunction is basically at the anaphase where they split, where those chromosomes split by the disappearance of this synaptonemal complex. Right, non-Mendelian factors are basically factors that genes that don't work according to the simple genetic Mendelian crossing that we usually do. So, because, for example, we could say that we could easily say that the gene for tallness is this and this, and then T and T, and then the organism that will form will be tall, the organism that will give rise to will be short. Those genes are not, they don't act independently. In fact, there are a number of factors that work together that come together to affect the behavior of a particular, the expression of a particular gene. Some other gene also affect the expression of a particular gene, and that phenomenon is called epistasis. We are going to talk about it before the end of this video, epistasis. We also have polygenic characteristics, especially for the gene for tallness, the trait tallness or height. Is a polygenic trait. It is not just the gene for tallness alone that affect that. There are environmental factors. There are the kind of food you eat. All of those things, they play out in determining what phenotype you will exhibit, and that is why they are called non-Mendel factors. Mendel factors are genes that work with the normal Mendelian law, while the non-Mendel factors are the ones that don't necessarily go directly with the Mendelian law. Then the locus is the particular portion of the chromosome. So, for example, if I have a chromatin strand here, and I pick this particular portion, and there is a gene there. This portion is called the locus. The locus, which is the particular portion of the chromosome where I can find a gene. The locus is a particular portion of the chromosome where I can find a gene. And that's basically it, the locus. So we are going to go to number 32 now. Number 32. I've decided to write more terminologies so that we can finish off the terms on time. We can exhaust the terms in time. The first one says, the number 32nd, we did locus last, which was, we said, is a particular portion of the chromosome that we can find a gene. Now, a nucleotide, remember, if you look at here, I wrote nucleotide and nucleoside. See, the difference is S and T, and they basically have their own peculiarity. Now, a nucleotide is composed of, is the simplest unit of, like the smallest unit of the DNA and the RNA. So, but they basically, so when it comes in double helix structure, I've told you before, it forms the DNA, and when it's just a single strand, then it is the RNA. But they also have a number of differences. Now you see the nucleotide basically contains what is called the nitrogenous bases, and the pentose sugar, and of course the third component. So these are the major three components of the nucleotide: the nitrogenous base, the pentose sugar in form of ribose or deoxyribose. I told you that, so you know that if it's deoxyribose, it will belong to what? DNA, and if it's ribose, it will belong to what? RNA. So deoxyribose means ribose sugar that has lost one oxygen molecule. So We have then the third component is the phosphate group. Now basically it is the phosphate group, if you remember, I said is the one that makes the component of the DNA to be acidic. So without this phosphate group, what we have is the nucleoside. So essentially, the nucleotide is a nitrogenous base, pentose sugar, and a phosphate group, while a nucleoside is one without the phosphate group. Hence, it is when we now covalently, bond this nitrogenous base and this to a phosphate group that will form a nucleotide. Nucleotide is basically nucleoside plus covalently bonded phosphate group to make acidic. Now, while the nucleoside is nucleotide minus the phosphate group, all right? Now, what are purines and pyrimidines? Remember I just talked about nitrogenous bases. Nitrogenous bases are basically they form the backbone of the RNA and DNA. Alright? So the nitrogenous bases are purines and pyrimidines. Now, purines are the ones that have the imidazole ring. They form the purines are basically pyrimidines and imidazole rings. Okay, so purines there are two types of purines in the RNA and DNA. We have the adenine and guanine. And this is based on their being complementary base pairs. All right, so you remember when we are doing DNA when they are DNA transcription, we have adenine pairing with G with guanine and thymine pairing with C or U. So these pyrimidines are the thymine, which is present in both RNA and DNA, or cytosine if we are talking about DNA, or uracil if we are talking about DNA. So cytosine is not present in RNA. Uracil is instead present, while the remaining three components are present. So that's basically the difference between purines and pyrimidines. Pyrimidines are these guys, and they are simple. They are called diazines. They are simple diazines. These three, the pyrimidines. While purines are basically pyrimidines plus imidazole rings. All right. Then the next one is euchromatin and heterochromatin. All right. The euchromatin is now notice the word eu. Eu means the word true. It's going to be the Greek word eu means true. So then chromatin, of course, from what we explained, is still the same. Chromatin is just the uncoiled strand of DNA in the cell. Uncoiled strand. So that uncoiled strand, when it is called eu, to show that it is truly chromatin, meaning that it is the uncondensed form. It is a form that is weakly, that is not perfectly stained, so it is not easily recognizable, and it occurs in the interphase stage, which is the dividing phase of the cell. So you remember that in the non-dividing, it is only chromatin that we have, where when replication takes place, that's the S phase of the interface. So it's the euchromatin that we have. But the heterochromatin, remember, look at the word hetero, which I told you will mean different, right? Chromatin is a type of chromatin in the cell that those ones, now these euchromatin are found, if this is the cell, this will be the nucleus. The euchromatin will be found somewhere in the middle of that nucleus, while the heterochromatins will be found close to the end of the nuclear envelope. This is the nuclear envelope. So the heterochromatin will be found here, nuclear envelope. Alright, so and they are more condensed than the chromatins. Of course, not as condensed as chromosomes actually, but they are more condensed than the euchromatin. And they are not. Now, the other difference between the euchromatin and the heterochromatin is that euchromatin are we call them to be transcriptionally active. You know, I talked about transcription here, so that is those base pairing, that is base pairing. So euchromatin are transcriptionally active because the enzyme that comes to open up that double-helix structure of DNA that I talked about in DNA replication is available to the chromatin. So is able to access the chromatin such that the RNA is able to transcribe whatever is in the DNA. So if it's transcribable, then it is euchromatin. While the heterochromatin is not transcribable. Another thing is that it is the euchromatin that contains what actually becomes the chromosomes which house the genes, so which actually controls genetic characteristics or traits of that individual, while the heterochromatin basically function for gene silencing. I think I wrote gene silencing somewhere. It basically functions for gene silencing and positioning of gene. So the gene silencing. All right. What gene silencing means is that genes that are not going to be functional in that individual, those are silenced genes. So you've explained it. Silencing means that it's going to make the gene non-functional, all right, non-expressed, all right. So that is the difference there. Now, what are histones? Remember we talked about histone protein. They are proteins that help to wind up the DNA into the cell. And I told you that the winding up is very essential because of the length of DNA. A DNA of an individual, I guess, is as long as I'll check that out later, but it's a very long structure and it's going to contain the code of a full individual. Imagine the code the amount of code that is working for me now, that code this, that resulted in this whole body. You can imagine. All right, so it has to be wound up so that it can be squeezed into that small structure, which is the nucleus. Now, so the histone protein are the ones that actually perform that function. They perform that function. And these same histone proteins, if you remember, we talked about chromomer. We said the chromomer are the portions that are bead-like in that chromosomal structure. So it is the histone protein that act in those portions to form higher levels of compactness. So that is histone proteins, wind up the chromatin. All right. Then karyotype is simply a diagram showing the number of chromosome sets and the shape of chromosomes that an individual will possess. So the process of doing is called karyotyping. Get the number of chromosomes and the shape of the chromosomes of an individual. All right. Now, metacentric. So we've gotten to terms that are associated with chromosome. We talked about chromosome. All right. So the terms that are associated with chromosome now are that karyotype and then metacentric. Now, metacentric means essentially we always see that the structure. Whenever we see the structure of chromosome, we always see something like X. Now, not all of them look like this X. Actually, not all of them look like this. Now, this is basically it. When the centromeric region, the centromere, lies exactly in the center, lies exactly at the center, it is going to be metacentric. Metacentric. Centromere. Meta, meaning meta simply means like something like in between two words or so. That is what the Greek word meta means. All right. So centromere. So when this centromere lies exactly between the two arms. These are called arms. These are called arms. Arms. Now the arm, you know, we denote it by the smaller arm being called P and the bigger arm being called Q. So usually we have P as the smaller arm, but because in the metacentric, so in the metacentric, the P arm and the Q arm are the same. All right. Now, but in the submetacentric, we have that now in the submetacentric, we are going to see a shape like this. Now notice in this structure that although it looks like this center is actually in between, but if you notice very carefully, you will see that this arm is quite shorter than this arm. Look at it. This arm is quite shorter than this arm. So this is going to be called submetacentric, meaning that the centromere is slightly displaced away from the middle. Just slightly displaced away from the middle. So that the P arm and the Q arm are now very visible. So they are now distinct. The P arm and the Q arm are distinct. Remember here, the P arm and the Q arm were essentially the same, but here the P and Q arm are now apparently different. So that's the submetacentric. Now in the subtelocentric is one in which the centromeric region is well displaced away from the center. So it's close to the end. Remember telo? I told you telo meaning ending, right? In telophase that we talked about in meiosis. Please do well to check that video if you haven't. So telo meaning it's like more displaced to the end than being in the middle. So the centromere is close to the end. So that is why it's called the subtelocentric. Then it is called telocentric if it is exactly at the end, almost exactly at the end, something like this. So if the centromere, that is, the centromere is essentially the point that joins two chromosomes together, all right? So if it is exactly at the end or almost at the tail end, then it is called the telocentric or the acrocentric. I told you acro means something that has been cut on the edge. So it looks like it has been cut, the arms have been cut on the edge. So here, the P arm and the Q arm are not even apparent at all. So that is the difference. Now, we also have the NF. NF basically stands for number fundamental. Number fundamental. I guess there's an E here. Now, what this is basically talking about is fundamental number. That's the meaning. Fundamental number. And the fundamental number of the chromosome of an individual, of a karyotype, of the karyotype of an individual, is the number of arms that it possesses. So basically, if you are metacentric, if the chromosome is metacentric or submetacentric or subtelocentric or telocentric, there's a way we designate numbers to them. So for example, here we believe that they have two arms, so we give them an NF of 2. Here we believe that they also have two distinct arms. These are two distinct arms, so we give them NF of 2. Why these ones? Because the arms are not very distinct, so we give them an NF of 1 and then an NF of 1. Remember metacentric, submetacentric, subtelocentric, and telocentric. So the subtelocentric and telocentric have a number fundamental of 1, and this one have a number fundamental of 2. So for example, for tilapia that has 44 chromosomes, so if it I'm just assuming, for example, if it has, let's say 20, or let's say it means that it has 22 pairs of chromosomes. That's the the meaning. So if it has, out of this 22, if it has, let's say 20 to be metacentric, so it means that the number fundamental for the 20 will be 2 times of 20, and that will be what? 40. Maybe the remaining 2 are in this group, then you have 1, 1, 1, 1, and that will be what? 42, the number fundamental. But of course, it's going to be higher than this. Essentially, it's going to be higher than this. So now, what are we saying? We are saying that the number fundamental is the number of arms designated for each of the chromosome in the karyotype. Alright, what's the genome? The genome is simply the whole genetic material that an organism carries. That's what you mean. Well, that's what we mean by genome. There's the total DNA sequence that turns to the chromosome, or chromatin that turns to the chromosome, of course. There's all of those together is what we call the genome of the individual. Alright, the interface. The interface is, in medicine, I did tell you that it stands for the difference between, a phase between 2 other phases. So when there is a division before, and then before the next division, the phase of maturation and differentiation of the cell is the phase of is the interface. Right? It's called the non-dividing phase of the cell, and it is split into 3 phases, which are the G1S and G2 phase. I covered what these are. I covered them in the meiosis video. Please make sure to watch that video. The next one is the prophase. The prophase is the longest phase of is the first dividing phase of the cell, and it is the longest, and it is essentially the preparative phase for the cell. Notice that even that we are told that P should stand for, P pro means before, so of course is in preparation for division. The metaphase, middle phase. So is the phase where the chromosomal pairs assemble themselves at the center of the spindle fiber. Alright? Then the metaphase, metaphase plate is basically the spindle fiber that are formed by the microtubules of the cell, the microtubules that are organized by the centriole. Alright, so we call them the metaphase plates. And metaphase plate doesn't necessarily just mean this. It's actually the way these guys orient themselves here. We call it altogether the metaphase plate. Alright, so of course, what we actually mean is this spindle fibers, but the way they orient themselves to is while they form the metaphase plate. Now the telophase, of course, telo, as I told you here, means what? End. So telophase is the phase of, and I told you in metaphase that they should, you can consider telo as to tear. So which means to divide, essentially. That is the cytokinesis. That's the phase after which cytokinesis occurs. Then asta. Okay, asta is gotten from the Greek word that means star. Alright, so star, the asta basically talk about the shape that the centriole forms in the centrosome. You know that when cell division wants to happen, the centriole are found in the centrosome. So the centrosome now begin to organize what we call cytoskeletons. The cytoskeletons are microfilaments, microtubules that are found in the cell. So the centriole essentially during cell division organizes these microtubules, organizes them in a way that at the polar ends of the cell, at those poles of the polar end of the cell, they form what is called an asta. Remember, if you watch that video, you're going to see me draw something like this at the end. So they form a star shape. So that is what we call an asta. The next one is tetrad, dyad, and monad. Remember, tetrad is basically the group of bivalents that are formed during the prophase, the essentially in the zygote stage or zygonema of meiotic one division. So this is called a bivalent. Go and watch that video so that this can be clearer. In the zygote stage, where synapses of this guy occurs, the chromosomes occur, they form bivalent. So the four arms are called tetrad. So that's why they work them as. Then dyad. In dyad is formed when separation has occurred. So after meiotic phase 1, which is a reductional division, a dyad is formed. This thing splits into the two chromosomes essentially. And the monad is formed after meiotic division 2, which is an equational division. So, and I told you why it is equational division, of course. So where you have your chromatid strands separately. Alright, so that is that. Then the homology surge. Homology surge is basically when in the leptotin stage or leptonema of the meiotic division 1, we said that homology surge is when the chromosomes that are homologous, that are supposed to be homologous pairs because they contain the same set of genes on the same portion of their structure, they are supposed to begin to find themselves, to attach to themselves. So that is the process of finding or looking for themselves is called the homology search. Then capturing. Capturing is what happens in metaphase. At the beginning of metaphase, the bivalent, or that is meiosis or homologous pairs in mitosis, will begin to move up to the metaphase split to assemble themselves. So once they get attached at this center and they are kineticals, then they are said to have been captured. They are said to have been captured. Alright, so and any microtubule extended there that doesn't capture a chromosome will essentially get removed or disappear. Alright, then the synaptonemal complex. Notice I drew a shape here. Is the complex that is formed between bivalent when they synapse. Alright? Then the hybrid and hybridization. Alright? So we've got this one looks like crossing here now. We have crossing. Crossing is basically, we could also use this side of the board since we have exhausted the terms here. Crossing is monohybrid cross and dihybrid cross. Crossing simply means the exchange of genetic material between the two parents. Alright, like when we actually draw a link between those genetic those gametes. Right, but let's get to hybrid first. An hybrid is an individual formed by the conjugation of individuals that have different genetic materials. So when I cross organisms of different species, I form what is called an hybrid. So that's what I mean by organisms with different genetic materials. So when I cross them together, I form what is called an hybrid. And the process of doing so is called hybridization. All right. Now, what is hybrid vigor? Hybrid vigor is, so we can say that hybrid is the product of outbreeding. Outbreeding. Yeah? Outbreeding. Because breeding within the same species is called inbreeding, and breeding out of the same species is called outbreeding. So hybrid is the product of outbreeding. Alright? Now, hybrid vigor simply refers to those characteristics that a vigor, vigor means vigor, if I say you have vigor, like the strength, the energy, that kind of thing, So the hybrid vigor simply refers to those characteristics that are possessed by that hybrid, the character that possessed by a hybrid that may help you to survive and thrive even better than the parents. Alright, so the features that this organism has that is more beneficial to it than that the parents from which it was gotten does not have. Those are the hybrid vigor. So, for example, after crossing some tilapia, you will see that you will notice that some will become more reproductively active, while others less active. You also notice some will have more growth than their parents. So that is an hybrid vigor. So in terms of growth, in terms of appearance, in terms of all of those characteristics, good characteristics that the hybrid has over the parent from which it's coming from, I want to call hybrid vigor. Now, heterosis is almost the same as hybrid vigor. In fact, it's the same, but it's just that heterosis is talking about a specific trait in which the hybrid has over a specific. You see, hybrid vigor is also called heterozygote advantage or outbreeding advantage. Yes. The hybrid vigor is called those advantages that that's the word. Those advantages that the hybrid has over the parent. So and those are the advantages that we're actually trying to get. That is why we have crossbreeding in between and outbreeded in our crossbreeding. Alright. Then so heterosis is basically about the one specific trait. Alright. Quercetin and sugar chain are basically proteins. So the quercetin are the ones that attach themselves between these bivalents, while sugar chain are the ones that hold sugar chain. I told you means protector in Japan. They are the ones that protect this quercetin from being attacked by separates. If you really want to understand that, just go and watch that Melis's video. Codominance now. Now, I think we're already beginning to talk about allelic interactions. So, in allelic interactions, we have what is called codominance. Now, codominance is essentially when two dominant alleles are expressing themselves fully in an organism. So, an example is the blood grouping AB in human, AB and O. So, you see that all of these three forming the AB blood group. Of course, AB blood group. So this is a codominance, meaning that both are completely dominant. They are showing they are both dominant and they are both expressing themselves phenotypically. While the incomplete dominance is what we call gene blending. That's gene silencing. Gene blending. I think I talked about it a while ago. In gene blending is incomplete dominance, such that, for example, if in Mendelian cross of a white flower and a red flower, what he derived was a pink flower, which was the color in between red and white. So that is gene blending or incomplete dominance. Both of them are trying to express themselves partially. It's also called partial dominance. Alright? Now, a complete dominance, on the other hand, is when one particular gene, one allele is dominant over another allele. Is completely dominant over another allele. For example, the trait for tallness usually might be dominant over the trait for shortness. Alright? So that is complete dominance of one gene. Then linkage. Linkage is basically talking about when two different genes occur together on a chromosome. That is it. That's linkage. So it is basically called linkage because of the high propensity for being delivered to the next generation. Those two genes, let's say for example gene A and B, gene 1 and 2, those gene 1 and 2 are so close, and you see this linkage is the genetic term that we use to check the closeness of the position, the relative position of. So when they are so close, that they get transferred to the next generation almost every time, then they are said to be linked genes. They are said to be linked genes. That they have a high propensity for being transferred together to the next generation. Alright, those are linked genes. Then mutation. So mutation is basically any alteration, whether permanent or temporary alteration in the genetic makeup of an individual, whether in the DNA or in the chromosome. They stem to mutation. That is why we have different types of mutation. We have the point mutation. We have the indel mutation. Indel mutation is just talking about insertion and deletion mutation. Right? It's a separate type of mutation on its own. And we also have the chromosomal mutation. Those are the ones we are going to talk about here. The chromosomal mutation. I think I wrote some of them there. And we also have the frame shift. We have the frame shift mutation. So we have indel, chromosomal point, and frame shift mutation. Alright. Now what is polyploidy? The next term, that is the number 65th term. Polyploidy, remember the word poly means many. So it simply means an individual with more than 2 sets of chromosomes. Remember we talked about haploid in meiosis. Haploid means haplous, which means single. Now, the diploid means 2, right? Diploid means 2 chromosome sets. Then polyploid will mean more than 2. And it could arise as a result of different reasons. So if you are, okay, so let's just treat this together. If you have 2 more sets of 2 chromosomes, that would be 4 sets of 2 chromosomes, it will be called tetraploid. Then if you have 3 sets of chromosomes, that will be called triploid. Alright? So and it basically caused by some different phenomena, such as when chromosomes undergo there's something called endoreproduction. You see, endoreproduction, endoreplication rather, or endoreproduction actually, endoreproduction. Please look that up. It basically means when a chromosome has been duplicated. The DNA has been duplicated in the interface in preparation for a cell division. Then the cell division now fails to happen. So if cell division fails to happen, then the organisms result in becoming a polyploid. Alright. Now, if this happens in the same species, organism of the same species, it is called. There are also some other types of mutation that will result in that, such as when there is translocation and some other, and there is non-disjunction can also cause that. Did I write non-disjunction? Okay, let's add it to number 87, for example. Number 87, we have non-disjunction. Alright, so if non-disjunction happens, it could also lead to polyploidy. Alright, so when you have more than 2 sets of chromosomes, that's the polyploidy. Then polygene, of course, poly means many as well, again, and gene, many genes. So polygene is talking about when more than 1 gene, 2, 3, or 4, or 5 sometimes, code for the same trait. Right? For the same expression. Right? Then translocation. You see the word translocation is going from 2 Greek words. We have trans and location. So trans meaning a shift in position. Of course, a shift in position. So how that happens is that when a chromosome shifts, trans means to move, location, to change location. All right. What basically happens is that a chromosome. So if I have a chromosome like this, for example, and basically a portion basically breaks off and moves to another, or maybe even two types. It depends on when two chromosomes actually joins together, move to another point, and they join to another chromosome. When the portion moves away and joins another chromosome that are not the same, that are not homologous pairs, then that's translocation. You also understand that. Let me explain the response. Insertion is this: when there is an addition, is a type of point mutation, that there is an additional. So, for example, the gene that is moved from a point to another point is translocated, but the chromosome that gets attached to, so let's say it gets attached to another point, let's say here, it gets attached to here, so this new chromosome comes in. Here, this one has undergone insertion, while this one has undergone translocation here. Alright? Then deletion is the permanent removal of a gene, a portion of the chromosome from the chromatin trans. So when the portion of the chromosome just leaves, that is deletion. That it doesn't return to that chromosome is called deletion. Then the inversion is this: when this portion, for example, of this chromosome rotates by 180 degrees, and still stays in that same position, occupies the same position, but it has rotated by 180 degrees, such that or even if it's just a portion of that nucleotide, such that the portion of that nucleotide, such that the base pairing, the DNA sequence, we have the 5' to 3' end. So that it rotates the configuration of that to like 3' to 5'. Alright? So although the gene will still be the same, but there's going to be an alteration in the form in which that gene will be expressed. Yes. Then so that is inversion. Then there is substitution. Substitution is a type of point mutation too. So we've done in there, that's inversion and deletion. Alright. Insertion and deletion. Then substitution. There are many types of substitution. Substitution, mutation. We have about 3 of them. I think of the substitution mutation, we have the silent substitution, and we have the silent substitution. We have the missense substitution. That's the name, and a nonsense. We have a silent, missense, and nonsense substitution. Alright, so a silent substitution, just know substitution. What substitution is basically is that a portion of the DNA or the nucleotide gets substituted by another. So when a portion of that DNA gets substituted by another, that is a type of substitution. But of course, nonsense substitution is when a DNA sequencing. I really needed to search that nonsense substitution the first time I saw it. It's just saying that when a portion of that DNA, when DNA replication is going on, there is a protein called codon that we're supposed to do that to codon. So codon is a non-replication protein that stops the replication of that protein. So if it gets initiated or synthesized prematurely into that sequence, so that the replication stops prematurely, it is called a nonsense substitution. Alright, then there is centric fusion. Centric fusion is basically, you see these two terms are the same, centric fusion and Robertsonian translocation. So Robertsonian translocation, Robertsonian centric fusion mean, you know, acrocentric that we talked about, acrocentric chromosomes. If an acrocentric chromosome translocates, that is move. I told you translation is movement of one chromosome to another chromosome. So when it translocates, when two translocate and they merge together, so these two chromosomes were coming. So this one is coming, this one was also coming, and they now met at one point, and they now eventually form two arms. It is called Robertsonian translocation. Note, it doesn't change their number fundamental, their fundamental number. It doesn't change it because the fundamental number of this one, when it was still an acrocentric, was 1, and this one 1, so it turns to what? 2. So it doesn't change. So that is what we call the Robertsonian translocation. And it occurs in gene 13, 14, 15, 21, and 22 of the human chromosome. So that is the Robertsonian translocation, when it occurs actually. Okay, I skipped this, gynogenesis, alright, so, or gynogenesis actually. Gynogenesis is the word gyno and gynogenesis got from 2 Greek words, which are gyne. Gyne is the Greek word for, is it Greek? Yes, I think it's Greek word for winning. Winning. I mean, a woman, of course. So, yeah, and genesis, of course, means to start up, to begin, to happen. So, what happens in gynogenesis is a kind of reproductive process too, where the individual, a new individual, simply arises from a female egg cell, a female gamete, without the actual syngamy of the male gamete with it. So when both male and female gamete do not fuse together and a new organism simply just arises from the female gamete, it's called gynogenesis. But of course, that process needs it requires the male gamete to also make it happen. Only that it is the production of the rise, that new individual that is arising, that is coming, or that is being produced, is not a function, is not a product of syngamy between the two, such that it is unaffected by the male gamete, by the content of the male gamete. So whether sex determination or whatever doesn't affect that new individual. So that's gynogenesis, simply arises from the female gamete. And the opposite of that will be androgenesis. Andro means male. So of course, when it happens that a new individual, there's certain organisms such as fish and amphibians, they undergo these two. So when a new individual arises from the male organism from the male gamete without the actual syngamy of both female and male gametes. But then homogamity, these 3 terms are for sex determination. So you see, what sex determination is basically talking about, which of the chromosome set determines sex in an individual. That's what we're talking about. So sex determination is the determination by the property that is contained in the gamete of individuals. So in humans, for example, the male determines the sex because of heterogamity. Male heterogamity, because our heterogamity in human is in male. Our female exhibits homogamity. By possessing XX chromosome, while male exhibits XY chromosome, Y being the smaller sized chromosome. So what happens is this: why it is the sex determinant is that if the X chromosome from the sperm fertilizes the egg, then it means that the individual is going to be a woman, a girl, a female. Then if the Y chromosome, of course, fertilizes the egg, then the individual is going to be a male. So Heterogamity determines sex, right? Then hermaphroditism. Hermaphrodite simply means an organism possess two reproductive organs. So, and there are different types of hermaphroditism in fish. We are going to talk about that later, please. Then the kinetochore. Kinetochore is just talking about, kinetochore is just talking about the portion of that, of the chromosome that gets attached. You see, there is a centromere. This is a centromere. The centromere simply means the portion of the DNA sequence where they join together, where those two chromosomes join together. But they are surrounded by what is called the kinetochore. So you know the magic. When I talk about this in meiosis, I said those microtubules are kinetochore microtubules because they are targeting the kinetochores of those bonded chromosomes. So that protein that winds it, that surrounds that centromere, is called the kinetochore. Alright, filial generation. The word fili means children in Greek. So filial generation means the set of offsprings that are produced. So if it's the first filial generation, means the first set of crossing you do, the kind of the set of offsprings that are produced will call your first filial generation. And then when you do another cross, you get your second filial generation, third filial generation, as you continue like that. And then crossing simply means, this is crossing, for example. This is crossing. So if, for example, I cross a heterozygous tall individual and a homozygous tall individual, they are going to produce gametes. When you want to cross, you have to first produce their gametes out. Note that you're going to produce their gametes out first. Don't cross directly from the parents. Produce their gametes out. So when these two cross, this is the very easy way to cross. You cross from here to here. You have T, that is the homozygous offspring. Then you cross from here again still. Now to the next guy. Then you have T and capital T again. Then here you cross from here to here. Then you have capital T and small t. The reason why we're writing capital T, let's not confuse you, is that in this case we are assuming that the tallness is dominant over the shortness. So it is a common practice to write the dominant allele before the recessive allele. So we can then cross this next so that it produces T sub T. So that does not mean that the shortness is never dominant. In some individuals, shortness is dominant. All right. So this kind of cross is called a monohybrid. A monohybrid cross. While the next, it's called monohybrid because we are crossing for only one trait, which is what? Tallness. So for example, we are crossing for skin color as well as this. Then it will be a dihybrid cross. So it means we are crossing for two different traits. Right? Then gene silence, I talked about that. It is a function of also gene positioning. Number 88, gene positioning. It is a function of, it's a function of the heterochromatin. So that's more condensed body than the euchromatin. What it does is, it is a site for silenced gene. What silenced genes are basically portions. Now listen carefully. There are portions of that DNA sequence that you find the chromatin, the histone proteins. The histone proteins actually hold up those genes in those positions that they cannot be replicated, that they are not available for replication. So thereby leading to genes being silenced. All right. So inbreeding is breeding within the same species or breeding outside of the species. Then non-disjunction. All right. You know that in the anaphase of meiosis, what happens is what? That those guys, chromosomes, homologous pairs, or maybe non-sister chromatids, will separate, non-sister chromosomes will separate, so they will split apart. That is called disjunction. The process is called. So when non-disjunction happens, is one of what gives rise to polyploidism. Polyploidism. So when it happens in the same species, it's called allopolyploidism. No, in the same species, it's called autopolyploidism. Why? When it happens by your own hybridization, when you are crossbreeding, then it's called allopolyploidism. That's what's different. Auto and allo. So when it happens naturally in the same species, within the same species, it's called, that's non-disjunction. When there is non-disjunction, I told you there are two things that lead to polyploidism. We have non-disjunction of those sister chromatids resulting in an individual that now has double the number of chromosomes that is supposed to have. So like having two sets of two chromosomes, that's 4 sets, tetraploidy. So the two things I said give rise to it are non-disjunction and endoreduplication, something like that. Endoreduplication, right? So endoreduplication and non-disjunction give rise to that. So because of a number of reasons, I'm going to continue. We have how many more terms to 100? We have 12 more terms. So I'm going to continue with 12 in my next video. So this is what I'll do. These are hermaphroditism in fish, and I'm going to talk about it as part of the terms in genetics. So about 4 of them, synchronous, petrandros, like that. We're just going to talk about many of all of those hermaphroditism. Alright, thank you very much for watching this. Okay, we've not ended it. I'm going to talk about the 12 later on. Thank you. I intentionally put this set at the last because I have made videos separately on some of them so that for better comprehension, because we'll only be talking about a little bit about them here because of time. So if you really want to understand them, the intricacies there, you have to go and check. You can go down in the description to check the videos that I linked there on some of these terminologies. All right. So, well, on number 89, it says epistasis. All right. The word epistasis is gotten from a Greek word, and it was coined by William Bateson, I think. All right. So, and it's gotten from the Greek word epistaph. Epistaph meaning standing over, or standing upon, or sitting upon. All right. Now, this is the idea in epistasis: a gene at one locus. So, a gene at a particular. Let me draw the chromosome such that it shows the centromere. Now, a gene at a particular locus. So a gene, let's say a gene at this locus and a gene at this locus. So let's call this gene A and let's call this gene B. So this gene A, if gene A is epistatic over gene B, it means that you see that they are not in the same locus, of course. So this gene on a different locus is having influence over gene B, and usually the kind of influence that it has is such one that makes it to mask this gene B, mask or sit upon it such that gene B is unable to express itself, or it determines how gene B is expressed. So when such happens, we say that gene A is epistatic over gene B. So we call that gene that is having that influence over the other, we call it the epistatic gene. The epistatic gene, while the gene that is being influenced is called the hypostatic gene. So we basically use this term, we use the term hypostatic, especially when gene A is acting in such a way as to suppress gene B. So if gene A suppresses gene B, then we say that gene A is epistatic over gene B, and gene B is hypostatic on gene A. So I made a video using Labrador to explain what basically happens here. And there are two forms of epistasis. We have the dominant epistasis, and the recessive epistasis. And it's very simple. It's just saying that if a gene is epistatic over another gene in its dominant form, such that if I have A or AA being epistatic over B in any form, as far as there is a dominant gene, I told you that we always represent the dominant allele by capital letter. So as far as there is just one capital letter, one dominant allele there that is suppressed, that is having influence over the other gene, then we say it's the dominant epistasis. But if a gene has influence over another gene in its recessive form, we call it a recessive epistasis. All right. So the next thing is aneuploidy. Now, most of this, aneuploidy is also gotten from two Greek words, which are and ploidy. Of course, ploidy is talking about the number of chromosome sets that an organism has, while anu is talking about is anu. When you have at the back of words, usually in Greek, it means abnormal. It means not. It means not. So anu means abnormal. So there are two sets. There are two forms of abnormality that occurs with chromosomes. It's either with the number, this total number of chromosome sets, or abnormality in some of the chromosomes. So, like so if it's the total chromosomal set, then it will be called polyploidy. It will result in polyploidy, so that the individual has more than two sets of, I've explained polyploidy, so that the individual has more than two sets of chromosomes, such as triploid, tetraploid, and the rest of that. All right. And we've said that in polyploids are not sexually, they don't, in humans, they don't exist because the individual will not survive. But of course, we have tetraploids in some other organisms, because of, such as fish, due to evolution. So, for example, the cyprinids, in the set of cyprinids family, we know that most cyprinids have chromosome number from between 48 and 60. So, but the common carp or goldfish has about 100 to 102 or 120 or so number of chromosomes. So what does that mean? It means that this thing has doubled, if not quadrupled. That is the total number of chromosomes. So we know that tetraploid or polyploidy exists. Some organisms exhibit that kind of chromosome set. But in aneuploids simply means abnormality in some of the chromosome units. So, for example, it could be that the chromosome, it's usually as a result of two possible things, which are non-disjunction. This is the most common reason for aneuploidy. Non-disjunction means at the anaphase, where chromosomes are supposed to separate. So if there is non-disjunction of a particular chromosome, such that one particular, such that when we now have our two cells being formed, a particular cell will now have more chromosome number than the other, then that is non-disjunction. Non-disjunction has occurred, and it could be for different reasons. It could be that the kinetochore is we said that at the Pachytene phase, we said that at the metaphase rather, or at the Pachytene phase, we said that it's at the Pachytene of of the prophets that the homologous pairs are going to pair up, and they will begin to move towards they are going to begin to move towards the metaphase plate. So at the metaphase, the metaphase would have formed this, the kinetochore would have formed a spindle fiber, and the spindle fiber will begin to begin to join to that kinetochore. So when they are joined, it's called capturing. So if capturing doesn't occur in that metaphase, there is a possibility of non-disjunction, such that instead of the spindle to go back with only a single set of chromosomes, instead of the particular spindle to go back with a single set of chromosomes, what you now notice is maybe the non-disjunction happened here. Then you will notice that this guy is going to go back with both chromosomes again. So you see, this one is going back with only one, this one is going back with only one, this one is going back with only one chromosome, this one is going back with only one, this one is going back with a set of chromosomes again. So and you're going to see the opposite side of the spindle, of course. This one will be going back with one chromosome, this one will go back with one chromosome, this one will go back with one chromosome, this one will go back to one chromosome. But here, there will be no chromosome going back here. So you see there is no disjunction here. So because of this, the organism, the cell that will be formed is going to have a +1 chromosome than this. One chromosome more than the second daughter cell. So that is why this form of aneuploidy is called there are two types of aneuploidy. We have the hypoploidy and the hyperploidy. Now, if the number of chromosomes reduces, such as monosomy and nolisomy, I've made a video about this, so you should do well to watch it. When the chromosome reduces, it's called hypoploidy, and when it increases, such as in trisomy and tetrasomy, it's called hyperploidy. All right. So basically, in trisomy, what you see is instead of have 2n, you're gonna in monosomy. What you see is instead of have 2n, you're gonna have 2n minus 1. Then in nolisomy, you're gonna have 2n minus 2. So meaning that two sets of chromosomes are disjointed, are not disjointed here. While here, only one chromosome is not disjointed, and so that's basically for aneuploidy. That's basically a particular set of, not the entire set, only some of the chromosomes there do not get disjointed. All right. Then the next term is pleiotropism. Pleiotropism is going from two Greek words, which are pleion and tropos. So pleion meaning many or more, while tropo from tropism, tropo means response. All right. So now what it means is that a particular gene has more than one influence, as it has influence on many traits. Okay, yes, that's the best word. That a gene at a particular locus, one locus, influences more than one trait. So a typical example is albinism in fish. The gene that codes for coloration or skin pigmentation. So if I name it A, let's name it A for example. If A is pleiotropic, it means that A is going to also determine other traits that phenotypically looks differentiated from each other. It will look like those traits are not even connected at all, and it is a result of one gene at one locus that is having influence like that. For example, in that, using that example, you are going to see that if a gene is recessive for this, you are going to see that gene is going to cause, so this will result in an albino. This is the normal skin pigmentation. So this will result in an albino. So an albino is going to have what? It's going to have no pigment on the retina, on the eye, non-pigmented eye. Then of course non-pigmented skin too. Non-pigmented skin. So and that is what. So you see, one particular gene at one locus has multiple effects. So that is what pleiotropism is. Then the next one is complementary genes. Two genes, let's say gene A and B, for example. You see there are different locus, different loci. So two genes A and B at different loci are said to be complementary if both of them must act together in order to fully exhibit a particular trait. For a particular trait to be fully exhibited by another, two of those gene A and B must work together. So we say those both genes are complementary. Meaning that in the absence of B, A will not give the full trait, express the full trait, and B will not, in the absence of A, B cannot express the fullness of the trait. Then supplementary gene is this. It's just like it's just almost like the opposite of complementary. Meaning that A is almost independent of A is independent of B, and B is independent of A. Only that if both of them are expressing almost the same gene, but almost the same trait, only that A can fully express the trait, but B only supplements it, only adds something to it. Then in the absence of A, B can also express the fullness of the trait, but in the presence of A, it only adds something to it. So that's supplementary gene. Then gene mapping. Gene mapping is basically used to, you remember when we talked about linkage, we said linkage is, so on a particular gene, on a particular chromosome, two genes are linked if they have a propensity for, I've talked about linkage in another video, and I'll link it down below. Two genes are said to be linked if they have a propensity, a greater probability of being transferred almost every time there is recombination. So if look at this gene now, look at this gene. What gene mapping is used to do is to check the relative position of two or more genes on a chromosome or in a particular locus, the distance apart, So that is what gene mapping is. So look at this gene now, this chromosome, these two genes, A and B. Notice that, of course, they are linked because they are on the same chromosomes. So as on the same chromosome. So as it is, if recombination occurs, there is a high probability that both of them will not be expressed in the individual together again. Why? Because they are far apart. They are far apart from each other. Unlike, look at a gene C here. So it means that if recombination occurs here with another gene, with another chromosome rather, if recombination occurs here and this gene recombines it and recombination occurs here, you will notice that it has missed this gene, but it has taken these two genes together. Why? Because their distance apart is not far at all. So the probability of passing them, bring them together again during recombination is high. So gene mapping is used to check for the relative position. Now, codon and anticodon. Codon is basically a nucleotide sequence that codes for a particular amino acid. So, and it's the one that triggers a stop or start for replication or for synthesis of a particular protein. Now, the difference between codon and anticodon is that codon is found on the messenger RNA. Remember when we did the dogma of life, we said that it is the messenger RNA that first goes to DNA to copy the code. So, and while anticodon is on the transfer RNA, and what it does is to check that if the codon has been coded for the right protein. Now the next term is penetrance. Penetrance is basically a measure of how a trait is being inherited among, how many offsprings inherited a particular trait. For example, if out of 100 offsprings, 80% exhibit a particular trait, say for example hemophilia, Then it means 80 of them then it means that the penetrance is in percentage, is calculated in percentage. So the penetrance of that trait is 80%. While expressivity is basically talking about in each individual that is in this among these 80 people, mean that 20% did not express that hemophilia. So in these 80% that express it, some express more than the other. So they express at different degrees. For example, some people were more severe than the other. So that severity is expressivity. It's talking about how much the intensity or the severity of that trait that is being exhibited. Then gonochorism. Gonochorism, of course, we have from the Greek word gono, which refers to gonad, of course, the sex organ or sex reproductive system. Then choros is actually talking about possession, right? Now, gonochorism is actually the other word that in plants, if two plants are differentiated in sexes, we call them dioecious. So, and the phenomenon is called, as gonochorism is called, dioecy. So it's just a term to show that a particular organism has separate sexes. That is an individual that is a male and an individual that is a female, separately. Lethal genes are genes that are capable of causing death to the organisms, but it depends on their combination. Usually when they come in homozygous forms, they are capable of causing death to the organism. So it's with respect to their combination anyways. So what they basically do is that they have the possibility of causing death to the organism that possesses them. Then hermaphroditism. Hermaphrodites are, okay, we've talked about hermaphrodites. We said they are organisms that, but basically what I want to talk about are the types of hermaphroditism. They are organisms that possess both male and female sex organs, alright, or sexual characteristics. And in fish, for example, there are 3 types of hermaphroditism that have been recognized. Of course, usually only talk about 2, and that is the simultaneous or synchronous hermaphroditism, and we have the sequential hermaphroditism. Now, in the simultaneous hermaphroditism is when, or synchronous hermaphroditism, of course, syn at the same time. Chronos is talking about time, while syn means same. So it means that the organism possess simultaneously, means happening at the same time. So it means that the organism possess both male and female sexual characteristics at the same time. Alright. While in sexual, and this is common with clownfish. While sequential hermaphroditism is when the organism possess one sexual characteristics first, and later in life it switches sex. So it switches sex, so that is sequential, that is one after the other. So for example, it could first be a male, then over time, after a while, it turns to a female, or starts with a female. So if it's a male before a female, it will be called protogynous. The female is called protogeny, while for the male it's called protandry. Andry basically refer to male, so gene refers to female. So protandry is when the organism will first be a male, then later in life switches to being a female. Then protogeny is the first. So you see, pro meaning before. There's to initially be a male, then switches to female. Initially be a female, then switches to male. Alright. Then there is also now I added this gel. In fact, there are 2 more terms that are on my mind that I will have to add. So, for example, I'm going to add the prezygotic and post-zygotic. So I had a question today, and the reason why I added this to, because I had a question today where someone was asking about if two organisms of different species, why they cannot give rise to a new organism. Alright, so I'm going to be answering that question with these two terminologies. Now, what is gel electrophoresis? In fact, the full name is actually agarose gel electrophoresis. Electrophoresis basically is coming from two Greek words: electricity, of course, and phoresis means moving. So it's basically talking about saying moving by electricity. And what is moving by electricity? It is a way of separating DNA molecules, DNA, into different fragments so that their sizes and charges can be checked. So the way the system is such that an electric field is set up in that system. So, and you know that the backbone of the DNA is actually made of phosphate, and phosphate possess a formal negative charge. Yes. So because they possess a formal negative charge, since there is an electric field being set up in the system, that electrophoresis system, what happens is that those DNA will naturally move toward, once the electric field begins, those DNA will naturally move towards the positive end of the plate. So their speed and how long, the distance they cover is used to check for their size. So and how do they check that? There is something called a gene ladder or something like that, in which comparison will be done based on the size of a DNA of known size, will be used to check the relative, to compare the distance moved by a particular DNA. So what gel electrophoresis is used to check is to check the size and charge of a DNA, to separate DNA based on size and charge. That's basically it. Alright, then prezygotic and postzygotic barriers. There are barriers so a prezygotic barrier will be that a barrier that comes before the zygote is formed. So basically, you see, there is something called nullisomy. You see, in nullisomy, one thing that has been discovered is that always the hybrid here that is formed, that is nullisomic, will not be sexually viable, will not be able to reproduce. Alright. But now, a prezygotic barrier is a barrier that determines, if it's a barrier, of course, it's going to stop. So it's going to hinder the zygote from forming. So because of prezygotic barriers, organisms of different species, the likelihood of organisms of the organisms of species that are very far apart from each other will not be viable because of prezygotic barriers that will come in place before even the zygote is formed. While post-zygotic barriers are what happens with nullisomy. Post-zygotic barriers form after the zygote has been formed. Those barriers hinder the reproductive capability of that hybrid. So those are prezygotic and post-zygotic barriers. Barriers that hinder either the viability of that hybrid at all, whether the hybrid can actually survive at all, or that actually stops the hybrid from being able to breed. Alright, so we talked about a number of things, and we are going to be stopping here. And we've talked about a number of terms, so I enjoin you to check on those videos that I told you about, so that you can really understand what each of these terminologies that I've talked about, what they mean in detail. Thank you very much. If you did gain value from the video so far, please do well to, that actually took me 2 days to make this video, so please let the labor not go to naught. Please do well to subscribe to the channel. God bless you.